lateral gene transfer between bacteria was understood. So they found that this extra chromosomal bacteria यहाँ से वहां जाता है. So what scientists tried was, inside this extra chromosomal DNA or plasmid, if you put in some gene from somewhere, they found that this also got transferred. This also got transferred and successfully accepted. Now, why have the reason I have included the next point is natural selection is, nature has this feature that whenever there is something new that is coming in, it will always accept, because it understands that it is going to prepare the organism for some upcoming danger. So what happens whenever we add something into the plasmid and put it inside some other organism, it was observed that it was taken up by the organism and incorporated into its genome. So this is the observation. Now, in this manner, people started doing what is called transgenic. They started generating genetically modified crops, genetically modified organisms, animals, everything. So in this panel, I'm not going through all of this because I think we have to look at gene-edited rice varieties right now. So I'm not going to the basics. Let us directly go into the insert picture here. So here you have a coin and the size of the cob. This was the native variety of corn years back. Now, when they genetically modified it, this is the amount of change that happens. You see how big the corn cob has become in comparison to the coin between the native variety and the genetically modified one. Whereas you have traditional methods of modifying. So many of the traditional farmers even now, do things like this, which is called a back cross. So from the so if you have two parents, so from the lower, the picture inset in the lower thing, you find that these two are the parents. On the sides, P1 and P2 are the parents. And what is in between, B, is the hybridized one. So here, though so by traditional methods itself, you can actually increase the size of the corn cob, but it is not as, dramatic as the previous one. So there is, so this is one thing I wanted to show, because there is not, in many cases, there is no requirement for a genetic modification, and we have still, gone ahead with it. So far we have tried to understand that all living organisms have a genome, which is made up of DNA, and that DNA has codes which get, translated into protein. And then we saw that the bacteria have DNA, which is not part of the genome, is what that extra chromosomal DNA, which is used for introducing DNA to another organism. So in fact, viruses also are used for transferring genes from one organism to another. However, I'm not going into that. We are just trying to understand literally, skim the surface literally. Now let us go and so we have understood what a genome is, how genetic modification happens, through that extra chromosomal DNA that we saw before. Now we will try and understand. Okay, suppose so I was mentioning that in that extra chromosomal DNA, you could add a gene of your choice. So whenever we have to add something, what would we do? That original plasmid, that extra chromosomal DNA. So you can think of it as a circle, okay, or a rubber band or something. Now, in that, what you would do, you would cut it open, attach the gene that you wish to attach, seal it, and then transfer it. No, so these would be the steps through which you would transfer. So that is how genes are transferred. So we would be looking at how it is cut. So why am I talking about this is, again, let us remember the dimensions that we are dealing with is so tiny, less than 1,000 times smaller than 2 lines of your fingerprint. In that dimension, we are doing these many modifications of cutting and sticking and so on. So I just wanted to bring let us look at that next. Now, before we go into that, let us look at genetic mutation. Now, genetic mutation again is one thing that we have always, we have heard since COVID times. So when we say mutation, it is when one thing changes or it, so how all you can mutate is given here. So you can have, this is the original message, for example, it says, I'll be by the lake. Okay, when you have a point mutation, you just change one alphabet. Suppose instead of THE, you say THW. So you get some information that is not able to, that give you the exact sense, but you are able to guess with reference to context. You can say, oh, maybe this is the only, with reference to context, you will be able to guess. When you say, suppose you change the alphabet lake to L to C, you will get the information changed to I'll be by the cake. So here information changes. So instead of, so this person who's probably trying to meet somebody, instead of going to the lake, they would probably go to a cake shop. You're, misleading that person. Next, you have something called a nonsense mutation where one alphabet gets deleted. What happens? The information itself, does not make sense if you just shift one alphabet from there. So I also mentioned about genome having the DNA only being made of 4 alphabets, and all of the protein is, the RNA and the DNA have their language consisting only of 3-letter words. So if I remove this alphabet, so here it says the cat and the dog. So the one in the lower panel, that is what is the DNA language. So it is GGG-AGC. Like, for example, if you delete A, because I told you it's all going to be 3-letter words, it will start taking this as the next word. Instead of AGC followed by CGA, it would become GGG, GCC, GAC. The whole thing will shift by one alphabet. That is what we call it as frame shift. So now that we have understood that, now I'm going into how we are going to prepare that plasmid for transferring our the gene that we want to introduce and put it into another one. So I was mentioning, right? So suppose it's like a rubber band. So let us imagine plasmid is like that rubber band. In that rubber band, you will have to cut at one place, introduce whatever you have to introduce, seal these ends, and then use it to transfer the gene that you have put in into another organism. So those that are capable of cutting exactly at some places are called nucleases, restriction endonucleases. So scientists found that something could cut in that dimension and precisely at some regions. So among the restriction endonuclease, you have enzymes which are this specific. So they can cut in such a way that it will have, it will cut it staggered. So it will have these ends which are sticky ends. So like in this wood, pieces of wood that you see, you have something hanging like that. So that is in the case of this particular example, BAM H1, if you see, it will cut here between the G's. So you will get something which is like this. This can be stuck. So for sticking, there is another enzyme. Or it can produce blunt ends like this. These also can be stuck. There are some places where you will get this kind of splinters also. Now, this splinter is what is significant in case of gene editing. So we'll quickly go through this video for us to understand how it is cutting, so that it'll help us imagine. So this is like your plasmid, the extra chromosomal DNA. So it is zoomed in now. This is the endonuclease that is coming in. It will go scan through the DNA to find the appropriate place where it will cut. And it will cut there. Now what is left is, staggered end. Now this is the gene that the scientist wants to introduce. That is introduced like that. And another enzyme will come and seal that place. That end also is sealed. So this is how the plasmid is modified and used for uh, transferring whatever modification we intend to incorporate. Okay. Now what happens in gene? Okay, this particular slide is going to tell us about how now we saw that something comes and attaches, right? So we saw that one enzyme went and cut the DNA, the extra gene was added, and something came and sealed the ends, right? So when the sealing happens, we have various ways in which the sealing will happen. So here, if the cut is like this, there are some enzymes which can perfectly align things and attach in a seamless manner. But there are some enzymes which will do a quick fix. So for example, suppose we have a plumbing issue at home. There might be, all of us would have had faced this issue. There will be one plumber who is very, very efficient, who would do a perfect job. But then there would be another person who will come immediately and fix it. So depending on the urgency of our situation, we would call somebody who, depending on how urgent the damage is or how urgently we need to fix it. So in case of gene editing, what happens is whenever this fixing happens, so I was telling you about this getting sealed, no? That sealing enzyme that switches on during gene editing is Cas9, which is actually a quick fixing enzyme. It is not the type of enzyme which will do a perfect job. So it will do something like this. If there is splintered wood, what will a carpenter do? You can actually, align it accordingly and then do it perfectly. Or you can completely shave it off and then attach it like a blunt end to end. So this particular enzyme that you see that is involved in gene editing does this kind of a job, like a quick fix. It removes parts of the DNA and then attaches it. So this is why we are worried about gene editing or any type of genetic modification, because whenever we modify, we are introducing extra genetic material inside. And when we introduce also, many of these methods actually clear off some of the DNA that is already present there or modify it even further. So what we would be probably generating would create a cascading effect. So in the beginning itself, I was mentioning about how the salivary gland would have all of the genes of the genome, but only those genes that are required for saliva secretion are turned on. The rest of them are turned off. Now, whenever we introduce something like this, when unintended modifications happen in the genome, what will happen is you are not sure what will get turned on unintentionally too. Or if something else that was originally supposed to be turned on is also getting turned off. So any of these modifications are, there is a possibility that such things can happen. So that is why we are against genetically modified organisms. So now I'm getting into CRISPR-Cas specifically. So I hope we are able to understand whatever we saw so far. We saw what is a genome, what is a gene. We saw how, how organisms, how the extra chromosomal DNA was used for transferring genes into another organism. Now we are getting it. So whatever we saw so far was called as transgenics. So under genetically modified organisms and transgenics, only these were included. This genetic modification using CRISPR-Cas, or this is now called gene editing. But from my understanding, it is only just wordplay. Even in gene editing, without modification of the genome, you can never achieve it. So this is to understand this, I would quickly take you to another video. I hope speed is okay.
Yeah, but now you've come to the critical part of genome-ed, so we'll continue at this pace.
So I'm directly taking you to what is CRISPR-Cas technology.
The CRISPR-Cas9 system is a tool.
So this actually, we're getting into the cell, the nucleus, again to the chromosome. So originally, this CRISPR-Cas technology was found to be the immune system of a bacterium, actually. So this particular protein that you see, this is what is CRISPR-Cas9. So CRISPR, the expansion of CRISPR is clustered, regularly interspaced, palindromic repeats. So in bacterial genome, scientists had found that there were genes which had interruptions in between, or there were repeats in between which there were some DNA. So when we say palindromic, so CRISPR, again, I'll go through clustered, regularly interspaced, palindromic repeats. So there are clusters of repeating sequences interspersed with something else present in the genome. And whatever is this repeating sequence is in the type of in the form of a palindrome. What is palindrome? Like for example, Madam, Malayalam, Nitin, all of these are palindromes, right? If you read from both sides, it reads the same. So you had such kind of repeats, and between them, there was some DNA present. When they checked what DNA it was, they found that this was DNA from a virus. So in a bacteria, so for a bacteria, virus is the one that will cause diseases. So it had information of the virus kept between repeating sequences, so that next time another virus came, it would check the DNA and it will understand whether whatever came in was a virus, has it affected us before, and then it will go and cut the virus's genome. So it was a kind of an immune system that the bacteria had. And that immune system had something called the Cas9 protein. So this protein would scan through the genome of, scan through any DNA that comes in, look for sequences which were, already available in its CRISPR, area. And if it was matching, it will go and cut it. So it was this protein that was exploited in CRISPR-Cas9 technology. Now, they found that this CRISPR had a RNA which was used for going and checking the DNA that was coming inside. So if it had this the same sequence, it will go and cut it. So what you see in gray, grayish blue is the one that will cut. This is the one that is used for checking. It's like your security check, for example. So if somebody has, so it's like a scanning machine. If that person has this ID card, they will pass through the biometric system and go in. It's like that. So this is the Cas9 protein. So this Cas9 protein is a cutting enzyme or restriction endonuclease. And it has this RNA which is going to guide it to that place where it has to get cut. This PAM is nothing but the palindromic sequence related one. See, it has found the place where it has to go cut. So the palindromic thing is like, so again, this is one example we can use to understand. So in a blind person's house, the house would be arranged according to step counts, right? So many steps will lead me to, say for example, chair. From here, if I take these many steps in this direction, I'll go somewhere else. Like that, the genome is organized. So the palindromic sequence is like the step count between the places where it has to get cut. Now here, this is the repair system. So here what happens once it is cut, our quick fix repair system, that which will remove the splinter, that will come and turn the gene off. So this is what is CRISPR-Cas technology. So what these people have done, whenever they claim that it is a gene-edited organism, they use one of these. So CRISPR-Cas, there are so many modifications of that CRISPR-Cas9 itself. Now you have Cas12 and so on. So these are modified proteins which have been trained to cut various they have changed, they have given us different guide RNA and so on. And they are using this protein, which actually comes from a bacteria, to cut any gene and modify any genome.
So
this is what is
Kritika,
gene editing technology. Yeah.
If you can go to what, yes, exactly, these slides
Yeah.
and
So
wrap up in.
what they have aimed at is, through this gene editing technology, they wanted to achieve genetically engineered, gene edited. So this is just wordplay. Any modification in the genome is a modification only. Like I mentioned, when you introduce a new gene also, there is, even though they say you have not introduced any other gene along with it, you've only gone and edited there. Like I mentioned, the repair mechanism is one that cuts off original DNA and then repairs. It's like that. Another thing that we have to remember, which I just want to mention here, is whenever whenever any modification is done, to identify those cells which have gotten this modification inside them, there is something called a marker gene which is always introduced. So so this is like, for example, there are a group of people, traveling somewhere. Some of them belong to one institution. They wear an ID or something, no? So this is like that ID card. So those organisms which have got this genetic modification incorporated inside are identified using a marker gene. This marker gene is a gene from outside only, no? So even in a gene-edited organism, there is no way that somebody can claim that extra gene is not introduced inside. So they have used gene editing to modify mushrooms to just change their color. They have used it to create, hornless cattle. Even human genome editing has been attempted in China, but thankfully this person got arrested. Nothing further has happened, but still, this is not a safe technology actually. So even though it is gene editing, even though they say it is precise, we understand that so many problems are possible. What all problems are listed in this are, they claim that it will be specific and it'll go cut only in that place. They found that that is not true. It goes and cuts randomly somewhere else also. Then this repair system, they say it's foolproof. That is also has been disputed. They said parts of the genome itself is eaten up. And non-targeted, so whatever genetic modification they targeted, that did not happen, something else happened, like the domino effect. All of these have been observed. So these are our potential points of concern. And another point of concern is what I mentioned right in the beginning, horizontal gene transfer. So any modification that we do, the risk of that getting incorporated into the human genome is also something that we are slowly understanding. So therefore, the need of the hour actually is regulation. So we have to regulation and creating awareness. So first thing, government has to have some regulatory systems in place where they would check what kind of modification is being done, why is it done, is there a route in which we can do it without genetic modification at all. Like for example, the hybrid variety that I showed you, there are so many native varieties of crops already existing where these desired things that are claimed, like for example, salt tolerance or drought tolerance. For these, there are so many native varieties which can be used. So
Yes, Shaumik
unnecessary
is covering that.
amplification is not required. So these are the regulations that we have to do. So thank you for your attention. Sorry, I exceeded a little bit, I guess.
No, thank you so much, Dr. Kritika. I think if I, as a layperson, have to sum up what you were saying, genome editing also needs a vehicle of a plasmid or an external vector, which can take the gene of interest into the host organism for genome editing to take place. Along with it go other foreign genes like marker genes and so on. That's point number one. Point number two, you are questioning the claim around precision, where they say that only the intended change will happen, no other change will occur. You are pointing out that the enzyme has the ability to make changes off target also, the protein that got introduced. And the fact that the foreign genes can get inserted inadvertently, though that was not the intention. So
Yes.
this whole narrative that GM is different, which is transgenic technology, had foreign genes used, whereas here there are no foreign genes. They claim that, yes, such inadvertent insertions happen, but we have cleaned it up. Afterwards, we checked, we did back crossing, we cleaned it up. But just to put the record straight, no one has seen those reports. But there's a lot of published literature that shows that inadvertently, even if it is not the intention, such genetic material does get inserted. Then we have evidence that shows that you didn't intend for some new protein to be produced. You thought you were doing something very precise as a geneticist, but the changes are so unpredictable and random, even in genome editing, that new proteins have been discovered, including through their phenotypic expression. That hornless cattle example from the USA and so on. So you're basically pointing out that it was like a primer. You tried to give a lot of simple anecdotes for us to understand how something like Cas9 or Cas12 operates, and then what happens with the repair mechanisms. And even in the repair mechanisms, what sort of randomness actually exists. Thank you so much for a very pictorial depiction of what is genome editing. Though it appears complicated, some of our participants have written saying that it's too scientific, too technical. I'm still not able to follow. That's all right. We are not going to be geneticists tomorrow. She gave us those basics so that we at least understand what we are talking about. But let's move to the next presentation, which is about the two rice varieties. And Shaumik will present the real world questions that emerge from what has happened with two rice varieties that have been genome edited. And I will say very, very intentionally that these two varieties are genetically modified, GM. Genome editing is not very separate from genetic modification. It's part and parcel of GM as per India's law, as well as when you understand the technique at the molecular level. Shaumik, I hope you are around and
Yes, yeah. Thank you. And good evening. I will note my presentation. Yes, is it visible?
yes, it is.
Yes. As we know that this gene-edited rice, or rather gene-modified rice, was released recently by our agriculture minister and the top officials of ICR. These two rices are named Pusa DST1 and DRR Dhan 100 or Kamla, and both of them are gene-edited. We will just look at their characters which have been published. So Pusa DST1 has been developed from a popular variety MTU 1010, which is already being cultivated in large area of India. And they have used the site-directed nuclease, that is SDN1 technology, as Dr. Kritika had just told you about nuclease, which are enzymes which goes and cuts DNA at specific points. So this is the SDN1 technology, which I will explain what it is. And this was developed in New Delhi, ICR, IARI. And this has a mutated form of drought and salt tolerance gene. So in the rice, there is a drought and salt tolerance gene, which has been mutated by using the gene editing technology. And by that mutation, they are claiming that the rice has got broader leaves and it has reduced stomata. And because it has reduced stomata, the water efficiency has increased because stomata is involved in transpiration. There are also several other functions, but they are focusing on the evapotranspiration. So when you have low stomatal density, or water efficiency goes up, and it has ability to withstand salt. And its yield is 2.4 to 3.7 metric tons per hectare in saline and alkaline soils, as
As
per
per the
their
claim.
trials. Yes, and it has
श्रमिक
been
आप थोड़ा
recommended
हिंदी
in.
में भी बताएंगे ना?
अच्छा अच्छा जी.
ये
तो
पूरा इंग्लिश में बताने के बाद थोड़ा हिंदी में भी बताइएगा.
जी.
धन्यवाद.
तो ये जो धान है पूसा DST राइस 1, इसको MTU 1010 एक पॉपुलर वैरायटी ऑलरेडी जो बहुत जगह में लोग लगा रहे, उसको जीन एडिट किया गया है. और इसमें साइट डायरेक्टेड न्यूक्लियस, जो कि शॉर्ट फॉर्म में SDN1 टेक्नोलॉजी यूज किया गया है, जिसमें अभी डॉक्टर कृतिका ने आप लोगों को बताया कि न्यूक्लियस एक ऐसी एंजाइम होती है जो DNA को एक स्पेसिफिक जगहों में काट देती है. तो इसको भी DNA को काटा गया है, कुछ जीन को, कुछ सीक्वेंसेस को निकाला गया है, और इसके जरिए इन्होंने जीन एडिट किया है. और इससे एक म्यूटेंट क्रिएट हुआ है, क्योंकि ओरिजिनल जो 1010 था वो तो बदल गया, क्योंकि उसके कुछ जीन्स हमने निकाल दिए. तो इसके चलते क्या हो रहा है कि उसके पत्ते चौड़े हो रहे हैं, उसके पत्तों में बहुत छोटे छोटे माइक्रोस्कोपिक छेद होते हैं, जिसको हम लोग स्टोमाटा कहते हैं, जिससे पानी वाष्प बन के आसमान में चला जाता है. तो ये इनकी कमी हो गई है इसमें. और कमी हो जाने के चलते वो कह रहे हैं कि इसकी जो पानी कम, माने कम पानी में भी ये धान हो जाता है. और ये नमकीन जमीन और क्षार वाली जमीन, अल्कलाइन जमीन में भी ये धान हो जाती है. और इसमें 2.5 से लेके 3.7 टन प्रति हेक्टर उपज आ रही है. और ये आंध्र प्रदेश से लेके आपका पश्चिम बंगाल तक जितने भी बड़े बड़े धान के राज्य है, उसमें ये वैरायटी रिकमेंड की गई है. शायद अगले साल या उससे अगले साल ये किसानों के पास पहुंचेगा. तो ये पूसा DST1 था. अब ये DRR धान 100 जो दूसरा धान इन्होंने निकाला है, इसको डेवलप किया गया है इंडियन इंस्टीट्यूट ऑफ राइस रिसर्च हैदराबाद में. इसका जो धान से इसको निकाला गया है वो है सांबा मसूरी. सांबा मसूरी भी एक पॉपुलर धान है, और इसको भी जीन एडिट किया गया. इसमें धान में एक एंजाइम होता है साइटोकाइनिन ऑक्सीडेज जीन एक होता है. बेसिकली साइटोकाइनिन एक हार्मोन है जो प्लांट ग्रोथ में बहुत भूमिका रहती है उसकी. उस साइटोकाइनिन को तोड़ने के लिए एक एंजाइम होता है साइटोकाइनिन ऑक्सीडेज. उस जीन को इन्होंने एडिट किया है. एडिट करने का मतलब वही, उसके DNA को काटा गया है और कुछ पार्ट्स निकाल दिए गए हैं. उसके चलते जो साइटोकाइनिन ऑक्सीडेज जो है वो उसकी जो काम था जो साइटोकाइनिन को तोड़ना, वो अफेक्टेड हुआ है, जिसके चलते ये क्लेम कर रहे हैं कि इसकी उपज बढ़ गई है, इसकी सूखा सहनशीलता मोटा मोटी एक है, मॉडरेट है, और ये जो है कम नाइट्रोजन में भी इसकी फलन आ जाती है। ये 20 दिन पहले इसकी मैच्योरिटी होती है। सांबा मसूरी लगभग 130 दिन में मैच्योर करती है, तो 110 दिन में ये मैच्योर कर जाती है, और इसमें 5.37 मेट्रिक टन पर प्रति हेक्टर यील्ड आ रही है। और इसका जो चावल है वो उसके जो क्वालिटी है वो सांबा मसूरी जैसा ही है, ऐसा वो क्लेम कर रहे हैं, और इसको भी कई राज्यों में रिकमेंड किया गया है।
English,
अभी हम लोग
Kamala English.
okay, sorry, I have to do in both lines. Some red mark is coming, I'm not able to understand. Anyway, so this Kamla is also known as DRR Dhan 100. It has been developed by the Indian Institute of Rice Research, Hyderabad, from Samba Masuri, which is another popular variety. And here also SDN1 gene editing has been used, but this is CRISPR-Cas 12A. The earlier PUSA DST was CRISPR-Cas9. This is CRISPR-Cas 12A, advanced technology from 9. And here, as we know, in plants, there is a hormone called cytokinin, and there is an enzyme called cytokinin oxidase, which breaks down cytokinin. So they have edited the gene which produces cytokinin oxidase, and there are different cytokinin oxidases in rice. They have basically edited the CKX2 gene, and by editing that they are claiming the yield has gone up, the drought tolerance, moderate drought tolerance is seen, and the high nitrogen efficiency is seen. The rice variety matures in 110 days, which is about 20 days earlier than its parent, and its yield is claimed to be 5.37 metric tons per hectare. The rice retains its grain cooking quality and is recommended for all these states which are similar, more or less similar to Busa DST1. Now, what is this site-directed nuclease? So there are three such technologies which are called SDN1, SDN2, and SDN3. Out of this, SDN1 and 2 have been exempted from, they are not considered gene modification. So what is the SDN1 technology which has been used to develop these two rice varieties? So SDN1 technology here, the double strand break is made, like Dr. Kritika had shown in DNA. There is double strand, two strands of this DNA, and then that is broken, that is cut. That is cut by what? By the CRISPR-Cas enzyme, that is the nuclease. So when it is cut, then certain genes are deleted in the process. And what happens is that the DNA again joins by the natural mechanism of the cell. And that is how the editing is done. So here what happens, there are changes in base pairs and certain parts of the gene are knocked out. So you actually cut the DNA at a specific place, and then the DNA again joins, and in the process some of the base pairs, that is what Dr. Kritika was showing, ATGC, some of those combinations are deleted and you get a new type of a DNA. And by this process, those changes which they have found, like the leaves are changing, the stomata is reducing, all those things are happening, and they have targeted it for a particular gene, that is the drought and salt tolerant gene. And in SDN2 also it is similar, but here there is a template which is also used for making specific changes. And SDN3, of course, that uses foreign DNA, so it is not under the exemption. It is normal gene modification. So what happened in 2022, the government actually exempted SDN1 and SDN2 type of tools from this genetic modification oversight. So they removed it from the Environment Protection Act 1986, and by this deregulation, so this deregulation was purposely done so that it opened the door for gene edited crops. And we can now see that two rice varieties have come out which are not under the oversight of the GEC because it is exempted. So once it is exempted, the safety and other procedures are much reduced. And 40 more crops are in the pipeline. So we will have more and more these type of crops coming up. Now with this gene editing technology, you can develop all types of products. You can develop different types of rices which may have more phosphorus efficiency or nitrogen efficiency or pest or disease tolerance or stress tolerance, or you can even create herbicide tolerance, and you can create different types of nutrients. So a lot of things can be done, and they have actually mapped out several such genes in rice which can be edited, and by editing those genes you can get different types of desired changes. तो इसमें 2022 में सरकार ने ये SDN1 और SDN2 जो दो टेक्निक है इनको एग्जेम्प्ट कर दिया, मतलब इनको जीन मॉडिफिकेशन के अंतर्गत नहीं माना गया। इसको माना गया कि ये इसमें कोई जीन मॉडिफिकेशन नहीं हो रही है, इसीलिए इनको छूट दी गई। और छूट देने के चलते ये 2 धान के वैरायटीज निकाले गए हैं और 40 और ऐसे फसल आगे निकालने का प्लान है। तो ये बेसिकली इसलिए किया गया ताकि जीन एडिटिंग टेक्नोलॉजी को लाया जा सके। नहीं तो पहले जो स्थिति थी उसमें कोई भी जीन मॉडिफिकेशन या जीन लेके कोई छेड़खानी करना अलाउड ही नहीं था, लेकिन इन दोनों टेक्नोलॉजी को एग्जेम्प्ट करके हमने इसके रास्ता खोल दिए और ये जीन एडिटिंग का काम शुरू हो गया, जो बिल्कुल जोरों शोरों से चल रहा है। अभी 2 वैरायटी निकले हैं और फिर और निकलते जाएंगे। और ये जीन एडिटिंग के जरिए आप लिटरली कुछ भी कर सकते हैं, अगर कोई चाहे तो कुछ भी कर सकते हैं। इस तरह का सिचुएशन आ रही है जिसमें आप किसी भी धान को सो कॉल्ड आप कोई कीट आ रहा है, वो आप कीट के आक्रमण से रोक सकते हैं, बीमारी से रोक सकते हैं, किसी का कुछ विटामिन बढ़ा सकते हैं, किसी का फास्फोरस नाइट्रोजन लेने की क्षमता को बढ़ा सकते हैं, किसी को नमकीन जमीन ठंड में गर्मी में कम पानी में, जैसे भी बहुत कुछ आप कर सकते हैं। और इसके लिए उन्होंने बहुत सारे जीन को टारगेट करके रखा हुआ है कि ये सब जीन के कुछ कुछ चेंजेस करने से ऐसे ऐसे हम लोग उपलब्धियों को ला सकते हैं। तो ऐसे देखने में तो ये बहुत अच्छा स्थिति लग रही है, लेकिन इसके अंदर बहुत सारे समस्या है, क्योंकि ये सारे जैसे कविता जी ने कहा ये लिविंग टेक्नोलॉजी है। इसमें जैसे हम एग्जैक्टली चाहते हैं वैसे ही नहीं हो जाता है, क्योंकि प्रकृति भी अपना कुछ कुछ कार्य करते हैं इसमें। तो पूसा DST
Sorry for interrupting. Can you stop sharing the screen and reproject it again? For the red lines, I have disabled the whiteboard, but still I'm unable to deal with the red lines.
Okay, I'll do that.
Sorry about it.
Yeah. Okay, so I'll go to that slide. So Pusa DST1 rice, how was it developed? Because here the paper is published in a journal, so we have come to know what they have done. So what they have done, as Dr. Kritika had told, that they had they have targeted the DST gene. DST is the drought and saline tolerant gene. So there is a gene in the rice which is having this property. So what they have done, they have created guide RNAs, and they have, because the guide RNAs cannot just the guide RNA will basically guide the Cas9 enzyme, the nuclease, to go to that site, and it would be cut at that particular site which the scientists want. So but the guide RNA can't go inside the cell just like that. It has to be packaged, it has to be put inside and something so that it can go inside, and that thing is the vector, which is the plasmid, which also was just talked about. So in the plasmid there the guide RNA is put and the Cas9 is put and they are attached, but even after that this plasmid cannot also get inside the cell. So it is further cloned into another bigger plasmid, which is the P. cambia 1300. Now this plasmid is used for agrobacterium transformations. Now agrobacterium basically is a bacteria which stays in soil and it creates plant tumors. It infects the plant and it creates some kind of swelling in the plant, which is just basically a tumor. So it goes and inserts it genetic material inside the plant. So that is why it is used for gene modification. And the same technology we are using in gene editing also, and yet we are saying they're different. Here also the agrobacterium technology is used. So what is happening? This guide RNA is cloned into the Cas9 complex, and then that is put inside the agrobacterium plasmid, and then the rice plants, the seeds are grown in a media. This is done in the laboratory, and in that media, when the rice is growing, this agrobacterium complex is put into that, and it infects the rice cells, and by that infection the genetic material is transferred into the rice and incorporated into the rice, and there is where the nucleus starts working. And once the nucleus reaches the particular site which has to be edited, the nucleus does its cutting work and removes certain base pairs from the DNA, and that is how we get the gene edited rice. After that, they do sequencing and they do screening, selection. Basically, they are saying that we are removing all these foreign materials. So what are those foreign materials? So we have the agrobacterium plasmid, which is a bacterial DNA which is going inside. Then there are antibiotic markers. So antibiotic markers like hygromycin, kanamycin, these are used so that for screening. So if the DNA, whether the DNA has got incorporated into the rice genome or not, is understood by giving antibiotics. So they will give hygromycin and all these antibiotics, and because that antibiotic resistance gene is there, so the cells which have got those genes, they will resist it, they will not die. The others which have not gone will die, and so that is how you will easily understand which have been transformed and which have not been transformed. So these are the marker genes, so these are also from bacteria. Then this CMV35S, this is a promoter. This is a cauliflower mosaic virus. CMV is cauliflower mosaic virus. So this one also is being used as a promoter, so that the gene, once it goes inside, it can express in a more vigorous fashion. So we have all these foreign elements going in. Of course, the scientists are claiming that we are removing them through screening, through selection, through PCR and all these technologies, but we are not sure because the information is not available. So what is happening because of this editing? So in the process of this editing, this cutting, 366 base pairs, so 366 ATGC, what Dr. Kritika was showing, A pairs with T and G pairs with C. So that is why it's known as base pair. So 366 base pairs are deleted in the drought and saline tolerant gene. So we are basically interfering with the DNA. We are removing several elements of the DNA, and we don't know what is their action. We are saying that we are talking of the positive results of it, but what are the negative results? We don't know, because nature had put those genes there. So and we are removing it, and we are saying there is only positive effects. That is something which is not possible. There may be some negative effects which we are not looking at. So when you delete those, so as Dr. Kritika said, that from DNA we get RNA, and then RNA translates into protein. So this again has effect on the protein. So in the protein also, what is protein? Protein is a big chain of amino acids. So there are large number of amino acids strung together like a garland. So from that 184 to 305, those amino acids in the series are not forming because you have deleted 366 base pairs. So the effect in the protein is also seen. And because these amino acids are deleted, then there is a new type of protein formed. It is not the same protein which the 1010 had. This is the mutant DST protein. and this mutant DST protein is actually leading to phenotypic changes. That is, you can see when you grow the rice, you see the rice has the leaf width has increased, the stomatal density has reduced, and this has resulted in its salt tolerance, and at the seedling stage, that is what they're claiming. So this is the positive part, but we don't know what are the other negative parts. So there is a loss of function. इसको हिंदी में भी बताएंगे. तो पूसा DST-1 जो धान चावल जो रिलीज की गई है उसको कैसे डेवलप किया गया, इसके लिए इन लोगों ने साइंटिस्टों ने एक गाइड RNA बनाया. गाइड RNA मतलब बेसिकली जो ड्राउट और माने सूखा और नमक सहनशीलता का जो जीन धान के अंदर थी उसको बदलना है, उसको चेंज करना है. तो चेंज करने के लिए पर्टिकुलर कुछ जगहों को उन्होंने माने पहले से निकाला है कि इस जगहों को अगर हम तोड़ देंगे, इसको अगर निकाल देंगे, हटा देंगे, तो ये कुछ इसका कुछ हमें लाभ मिलेगा. तो उसको न्यूक्लियस को वहां लेके जाना है, वो जो कैंची है, जो एंजाइम है, उसको उसी जगह में लेके जाना है. कैसे लेके जाएंगे, तो उसके लिए एक गाइड बनाया. जैसे हम लोग कहीं जाते हैं, कोई जगह के बारे में नहीं पता तो किसी को गाइड रखते हैं कि भाई मेरे को थोड़ा रास्ता बता दो. तो वो न्यूक्लियस को अब वहां जाना है, इतना बड़ा DNA है, उसको गाइड करके वहां पे ले जाने के लिए उन्होंने गाइड RNA बनाया है. और गाइड RNA फिर अकेले गाइड RNA और ये न्यूक्लियस अपने आप सेल के अंदर नहीं घुस सकते, तो इसके लिए एक ढांचा बनाते जो कि एक प्लाज्मिड एक ढांचा होता है, एक एक चीज होती है, एक वाहन होता है, जिससे कि वो उसको वहां अंदर सेल के अंदर लेके जाएंगे. और उसके लिए उन्होंने एक बैक्टीरिया इस्तेमाल किया है, जो एक बैक्टीरिया जब मिट्टी में रहती है, एग्रोबैक्टीरियम कहते हैं. वो पौधों के अंदर एक इन्फेक्शन क्रिएट करता है, एक ट्यूमर जैसा इन्फेक्शन क्रिएट करता है. ट्यूमर मतलब एक फूल जाता है पेड़. तो वो उस बैक्टीरिया के अंदर इस गाइड को घुसाए है, गाइड RNA को घुसाए है, न्यूक्लियस को घुसाए है, और उस ढांचा को उन्होंने चावल के जो बीज है उसको लैब में उन्होंने उगाया है एक मीडिया में, और वहां पे वो इन्फेक्शन कराया, वो संक्रमण कराया, और संक्रमण के जरिए वो जेनेटिक मटेरियल, वो जो ढांचा है वो आपका धान के सेल के अंदर चला गया. वहां जाके वो उसको जो काम करना है, उसको DNA को काटना है, उन्होंने वो काम किया है, और उसके जरिए हम लोग को जीन एडिटेड धान मिला है. अब देखिए इसमें कई सारे बाहरी चीजें हैं. सरकार कह रही है कि बाहरी कोई चीजें नहीं, फॉरेन चीजें नहीं है, लेकिन इसमें एग्रोबैक्टीरियम का जीन इन्वॉल्व है. इसमें जो हमारा एंटीबायोटिक मार्कर्स इन्वॉल्व है. एंटीबायोटिक मार्कर का मतलब क्या, अब वो अंदर गया है कि नहीं उसको जानने के लिए वो एंटीबायोटिक मार्कर जीन इस्तेमाल करते हैं. मतलब एक आई कार्ड जैसा है, वो बताते हैं कि वो जीन अंदर गया है कि नहीं. तो एंटीबायोटिक इस्तेमाल करते हैं. एंटीबायोटिक से अगर सेल मर जाता है, इसका मतलब उसमें कुछ गया नहीं है, और जो सेल जिंदा रहता है, इसका मतलब उसके अंदर जीन गया है. तो एंटीबायोटिक मार्कर जीन जो बैक्टीरिया से लिया जाता है उसको यूज किया है उन्होंने. उन्होंने एग्रोबैक्टीरियम के जीन इस्तेमाल किए हैं. उन्होंने एक प्रोमोटर भी इस्तेमाल किया है. मतलब जीन अंदर जाके ही ऐसे काम नहीं करने लग जाते, उसको थोड़ा सा प्रोत्साहन करना पड़ता है. उसको प्रोत्साहन करने वाला जो होता है वो प्रोमोटर होता है. वो कौन है, वो आपका जो फूल गोभी में एक वायरस लगता है उसका एक पार्ट उन्होंने लिया है. तो तरह तरह की चीजें अंदर घुसे हैं. फिर साइंटिस्ट लोग कह रहे हैं कि हमने सबको साफ करके निकाल दिया, फिर अंदर और कुछ नहीं है. लेकिन अब इसको मानना, इसको फिर देखना पड़ेगा कि ये सही में इस तरह है या नहीं है. कि उन लोग कह रहे हैं, लेकिन हमारे पास कोई ऐसे पब्लिक के पास कोई रिपोर्ट नहीं है कि ये सब बिल्कुल साफ हो गया, कुछ नहीं है. उसके बाद इससे क्या हुआ, ये सब करने से इससे क्या हुआ कि 366 जो DNA के बेस पेयर्स थे, मतलब एक दूसरे से जुड़े हुए जुड़वे कुछ चीजें होते हैं DNA में, उनको उन्होंने तोड़ दिया, उनको निकाल दिया. 366 ऐसे सीरीज में जीन का जो एक पार्ट था उन्होंने उसको निकाल दिया. अब हम जानते हैं कि जीन से ही प्रोटीन बनती है. अब वहां निकल गया तो प्रोटीन में भी निकल जाएगा. तो प्रोटीन में भी हमारे जो है करीबन 120-130 प्रोटीन के अंदर एमिनो एसिड्स होते हैं. तो प्रोटीन को बनाता है एमिनो एसिड. तो प्रोटीन के जो ईंटें होती है एमिनो एसिड होते हैं, तो 130 ईंटें वहां से गायब हो गए। तो इसके वजह से वो जो नया प्रोटीन बना, वो नया प्रोटीन को हम कहेंगे म्यूटेंट प्रोटीन, और उसके चलते पत्ते चौड़े हो रहे हैं, स्टोमाटा घट रहे हैं, और उसके चलते हमारा सॉल्ट टॉलरेंस और ये सब चीजें हो रहे हैं। तो ये बेसिकली उन्होंने किया है, उन्होंने पूरी तरह डीएनए के साथ छेड़खानी की है और उसको चेंज किया है, उसको काटा है, उसमें से कुछ चीजें निकाल दिए हैं, और हम लोग उसके जो अच्छे परिणाम है उसके बारे में बात कर रहे हैं। लेकिन उसके जो नेगेटिव परिणाम है, नकारात्मक परिणाम क्या है, उसके बारे में हम लोग को अभी तक पता नहीं है। अब
हम लोग
दूसरी जो
नहीं,
धान,
वे लोग सिर्फ पॉजिटिव बातें ही कर रहे हैं।
जी, now coming to DRR Dhan 100, which is also known as Kamla, we don't have much information about this because the paper is also not published. Research paper on this variety is not yet published. So whatever material we could get. So as I said, here cytokinin is a major plant hormone which is involved in regulating cell division, leaf development, shoot growth. So it has a lot of functions. And there is an enzyme called cytokinin oxidase, which is CKX for short, and they basically regulate cytokinin. So whether there should be more cytokinin or less cytokinin, that is regulated by this enzyme. So through gene editing, they have actually actually edited the gene responsible to produce that enzyme. Now in rice, there are 11 cytokinin oxidases, out of which the cytokinin oxidase 2 gene has been edited. Not all of them have been edited, only the 2 cytokinin oxidase gene has been edited. So in that gene, in that cytokinin oxidase 2 gene, there is a particular particular segment, which is the GN1A. And this, from earlier research, they have found that this is associated with high grain number, higher panicles, and lodging resistance. This research is available from earlier times. So they have basically edited those parts. So here they have used CRISPR-Cas 12A instead of Cas9. So they have edited 2 exons in the Samba Masuri, that is the parent, and this has led to 16 base pair and 6 base pair deletions in exon 1 and 11 base pair deletion in exon 3. So 2 exons have been edited where you have got these deletions. So it is same, again they have knocked out certain segments of the gene. And they here also they have used CMV35S. This is the cauliflower mosaic virus promoter antibiotic resistant marker genes, and they have used the similar technology, but we don't have all the details. So basically it is the same type of technology where they have identified certain genes, they have knocked out segments of that gene, and because of those knocked out segments, the protein is also affected, and the protein is not able to do its function as it would have done naturally. And that has resulted in some positive effects which are being highlighted, and we are not aware about what are the negative effects, or maybe the negative effects are not being evaluated, or they are not showing now, they may show some years down the line. So we don't have all those information. So now अब जो अच्छा इसका हिंदी, तो DRR धान 100 ये दूसरी धान है। इसको कैसे डेवलप किया गया, इसमें एक पौधे में एक हार्मोन होता है साइटोकाइनिन, जो पौधों का विकास, उसका पत्ते का विकास, उसका बढ़ोतरी, ये सब में बहुत मुख्य भूमिका रहती है। उसको रेगुलेट करने के लिए, अब वो ज्यादा हो जाए तो कुछ ज्यादा, बहुत ज्यादा बढ़ जाएगा, वो भी दिक्कत है, बहुत कम रहेगा तो कोई बढ़ेगा ही नहीं। तो इन कितना मात्रा में होना चाहिए या नहीं होना चाहिए, इसको मैनेज करने के लिए एक और एंजाइम होता है, उसको बोलते हैं साइटोकाइनिन ऑक्सीडेज, जो साइटोकाइनिन को तोड़ देता है तो वो काम नहीं कर पाते। तो अभी उन्होंने क्या किया है कि साइटोकाइनिन ऑक्सीडेज जो एंजाइम है उसको बनाता है, साइटोकाइनिन ऑक्सीडेज का एक जीन भी है, उस जीन से फिर प्रोटीन बनती है। तो उस जीन का कुछ सेगमेंट को उन्होंने एडिट किया है। तो उसको उन्होंने एडिट कैसे किया है, CRISPR-Cas12a का इस्तेमाल करके। पिछले बार हमने बताया था CRISPR-Cas9, ये उसका एक एडवांस्ड वर्जन है। यहाँ पे उन्होंने एक जीन का पर्टिकुलर एरिया को टारगेट किया है और वहाँ पे उन्होंने कई सारे उसके जो बेस पेयर्स है, जीन के कुछ हिस्से हैं, उनको उन्होंने हटा दिया है। और उसको हटाने के चक्कर में जो है उसकी उपज बढ़ गई है, उसकी सूखा सहनशीलता थोड़ा बहुत बढ़ गई है, ये सब वो क्लेम कर रहे हैं। इसमें भी उन्होंने एंटीबायोटिक रेजिस्टेंस मार्कर इस्तेमाल किया है, CMV35S प्रोमोटर इस्तेमाल किया, जो पिछले बार भी मैंने बताया था, जो फॉरेन जीन्स है इसकी भी इस्तेमाल हुई है। और इसके बारे में हम लोग को बहुत ज्यादा नहीं पता, क्योंकि इसका जो रिसर्च पेपर है वो भी पब्लिश नहीं हुआ है। तो रिसर्च पेपर से ही और जानकारी मिल सकती थी, तो जो अभी हमारे पास नहीं है। अब ये जो दोनों जीन को इन्होंने उड़ा दिया है, मतलब उसके कुछ हिस्सों को, तो ये जीन कई सारे काम करते हैं पेड़ में, पौधों में। और ये जैसे DST जीन है, वो तो सब जानते हैं कि ड्राउट और सॉल्ट स्ट्रेस को मैनेज करते हैं। इसके अलावा सेल मेम्ब्रेन जो होता है उसका प्रोटेक्शन करते हैं, सोडियम पोटेशियम आयन से उनकी बैलेंस को मेंटेन करते हैं, स्टोमाटा कब खुलेगा बंद होगा उसको मैनेज करते हैं, स्ट्रेस रिस्पॉन्स के लिए एक हाइड्रोजन पेरोक्साइड मॉड्यूलेशन होता है, उसको भी वो मैनेज करते, पानी कितना रुकेगा या उसको निकाल दिया जाएगा उसके ऊपर काम करते, कई सारे उसके फंक्शंस हैं। जब आपने इसको कुछ इसके हिस्सों को उड़ा दिया, अब उसका आप कह रहे हो कि बहुत अच्छे अच्छे रिजल्ट आ रहे हैं, लेकिन उसके कुछ गलत परिणाम भी हो सकता है, क्योंकि इतने सारे फंक्शन है। क्या हम लोगों ने हर फंक्शन को स्टडी किया, क्या हम लोगों ने हर चीज को देखा कि वो क्या बदलाव ला रहा है या नहीं ला रहा है? और इसको देखने में शायद कई समय भी बहुत लगेगा। इसके बाद अगर हम CKX2 जीन को देखें, जो दूसरी वाली कमला में जो चेंज की गई है, तो उसके भी बहुत सारे फंक्शन है। यहाँ पे आप देख सकते हैं कि उसमें प्लांट हाइट भी है, उसमें स्ट्रेस टॉलरेंस है, फॉस्फेट डेफिशिएंसी टॉलरेंस है, स्टार्च एंड प्रोटीन कंटेंट है, मतलब जो हम खा रहे हैं उसमें कितनी प्रोटीन कितनी स्टार्च आएगी, ग्रेन की एमाइलोज क्या होगी, ये सारा चीज इसके ऊपर डिपेंड करता है। अब हमने उनको उनको कुछ कुछ उसका हिस्सा को चेंज कर दिया, हटा दिया। अब उसका क्या फल इन सब चीजों में पड़ेंगे, इसके ऊपर अभी तक कोई स्टडी नहीं हुई है। थोड़ा बहुत स्टडी उन्होंने किया है, लेकिन वो मेरे ख्याल से सफिशिएंट नहीं है, और जो किए भी है वो भी किसी को पता नहीं है, क्योंकि कोई चीज पब्लिक के पास नहीं है। तो इस तरह से उन लोगों ने ये बदलाव लाया है। अब अगर आप देखेंगे, ये ऊपर कोने में जो है, बायां साइड के कोने में, ये नॉर्मल GM टेक्नोलॉजी है, जिसमें भी हम एग्रोबैक्टीरियम यूज करते, जिसमें भी हम एग्रोबैक्टीरियम के जरिए फॉरेन जीन को अंदर घुसाते, फिर उसका ट्रांसफॉर्मेशन करते, फिर उसको उगाते और उसका फिर कैरेक्टर्स देखते। यहाँ पे भी हमने वैसे ही चीजें इस्तेमाल की, यहाँ पे भी हमने एग्रोबैक्टीरियम यूज किया। लास्ट में जाके हमने थोड़ा सीक्वेंसिंग सिलेक्शन किया, जो कि हम कह रहे हैं कि हम फॉरेन जीन को हटा रहे। मतलब बेसिकली कहने का मतलब ये है कि हमने सब सेम सेम चीजें यूज किया, सेम सेम प्रक्रिया यूज किए, लेकिन एक को हम कह रहे जीन मॉडिफाइड, एक को हम कह रहे जीन एडिट, लेकिन दोनों का मूल स्वरूप एक ही जैसा है। तो ओके, so if we look at the functions of DST gene in rice, so there are several functions apart from drought and salt stress. So they are involved in osmotic balance, they are involved in stomatal regulation, they are involved in water retention, growth. Similarly, CKX2, which is cytokine oxidase 2 gene, that has also several functions, including functions of starch and protein content, including phosphate deficiency tolerance, crown adventitious root growth, plant height, nitrogen uptake. So several functions. These are pleiotropic genes, means they have multiple actions in the plant. It is not that I go there and change something and only that will happen and nothing else will happen. So we need to actually check what are the effects in these so many different things. Scientists are claiming we have checked, but we don't know what they have checked and what they have not checked and what they have just ignored. So all those things are not available to the public. And if we look at the two technologies, the tools that has been used, they are more or less the same. Even in gene modified, we use agrobacterium for cell transformation. We use antibiotic marker genes. Here also we are using those things. We are also using cauliflower mosaic virus promoter. There also we use, and yet the government is telling this is safe technology. This does not involve foreign gene. This nothing is there, but we are not sure whether they are there or not there. And then there was a rebuttal that basically gene editing and mutation breeding are the same thing, but they are not the same thing. They are not equivalent, because even if the gene editing is considered specific, which is of course more specific than mutation breeding, but still there are off-target effects. It is not that where you will send the nucleus, it will go and work there only, because after all these are human technology. Okay, we are improving, maybe after much research people may come with such things, but right now the technology is not foolproof, and it there are several off target effects, and there are several research papers on that. And in mutation breeding we are using some chemical or radiation from outside the plant or the cell, we are giving that. Here we are directly going inside the nucleus and changing the genomic materials. And that way we are actually bypassing the natural gene regulation, because we are sending nucleuses, they are randomly going and cutting different things, different places, and accordingly they're making the changes. So the natural process of gene regulation is bypassed here. Foreign genes are definitely involved, which we have already talked about. Antibiotic resistance is also involved. And here, because it is not precise, whatever is similar looking, so there are several similar looking gene clusters, and all those clusters can get targeted. It is not necessary that only GN1A particular 5 prime untranslated region of particular rice that will only be targeted. There could be similar sequences which are similar looking, and there the nucleus can go and do its work. Then there are certain segments of the genome which are protected. Which are protected means they have very important regulatory functions, and nature protects them from mutations. Mutations are always happening, but even then those protected sequences are not mutated because they're very critical to the organism. But here, because the nucleus does not know which is protected and which is not protected, its job is to see the sequence and cut it anywhere it sees it. It may go and cut it there. So in the normal mutation breeding, this will not happen. And every gene has at least 2 copies or more than 2 copies in the plant or in the humans or whatever. So in case of mutation breeding, maybe 1 copy is changed, but the other copy is still retained. So if the plant wants to go back to its original self, it can, because the origin 1 copy is still there. Here all the copies are changed, because the nucleus will not stop its activity till all the copies are changed. So this is an irreversible process. It is completely changed forever until you again do another gene editing to come back to the original self. It cannot reverse itself. And so it basically leads to substantial changes in the plant's phenotype, which we have already seen. And because the DNA is changed, is it the same 1010 or is it the same Samba Masuri? It is something else. It is a new plant. Even it's different from many other paddy varieties, because other paddy varieties will have all those base pairs. This one doesn't have. And here the changes are smaller in size, but they are more numerous. So there are a lot of differences between conventional mutation breeding using chemicals and irradiation and gene editing. So they are not equivalent, and government says that they are equivalent, and we did not have any problems with mutation breeding. Why we are having problems here? Because it's not the same. And so there is no question of substantial equivalence. And after all this, why do we need all this? Why do we need? We have a lot of stress tolerant rices. We have a lot of high productivity rices. We can use them. We don't need to go for these type of risky technologies. तो यहाँ पे सरकार कहती है कि म्यूटेशन ब्रीडिंग, जो कि बहुत पहले से ही किया जा रहा है, जिसमें कोई रसायन या कोई मतलब रेडियो एक्टिव रे इस्तेमाल करके पौधों को बदला जाता था उस केमिकल के वजह से या उस रेडिएशन के वजह से। तो वो कह रहे हैं कि उसमें तो किसी ने अपोज नहीं किया, तो इसमें क्यों कर रहे, इसमें भी तो एक्चुअली हम लोग म्यूटेट कर रहे हैं इसको। तो वो कह रहे हैं कि दोनों सेम है, तो आप
समित
उसमें
थोड़ा
कुछ
क्रिस्पली ट्रांसलेट करेंगे, शायद टाइम
जी
की इससे देख रही
जी
हूँ,
जी।
सॉरी।
तो वो बेसिकली दोनों बराबर नहीं है। इसलिए बराबर नहीं है क्योंकि इसमें हम लोग बिल्कुल जीनोमिक लेवल पे चेंजेस कर रहे हैं, और जो चेंजेस हो रहे हैं वो सारे जगहों पे हो जा रहा है। जहां जहां वैसे वैसे उनको सिमिलर सीक्वेंसेस मिलेंगे, जहां जहां उनको दिखेगा कि वो मिलता जुलता चीज है, उसको वो चेंज कर देंगे, उसको वो काट देंगे। उसके बाद इसमें फॉरेन जीन भी इस्तेमाल हो रहा है, जो कि हम लोग देख लिए कि बैक्टीरिया के जीन इस्तेमाल हो रहे हैं, और इसमें सारे कॉपीज को चेंज कर दिए जा रहे हैं। जितने भी कॉपी जीन के कम से कम 2 कॉपीज होते हैं हर ऑर्गेनिज्म में, उन दोनों कॉपीज को इसमें चेंज कर दिए जा रहे हैं। तो इसके चलते क्या हो रहा है, अगर कभी पौधा वापस जाना चाहता है उसकी कोई गुंजाइश नहीं है, क्योंकि सारे कॉपीज चेंज हो गए। और इसमें बड़े बड़े चेंजेस तो नहीं हो रहे, लेकिन छोटे छोटे बहुत सारे चेंजेस हो रहे। तो इसका मतलब ये हुआ कि दोनों एक बराबर नहीं है, जो गवर्नमेंट क्लेम कर रही है। अब बात उठती है कि इतना कुछ करने की जरूरत क्या है, ये जीन एडिट करके इसको निकालना, फिर उसके इतने सारे टेस्ट करके ये सब करना, इसकी जरूरत क्या है। आखिर हमारे पास ऐसे बहुत सारे धान है, ऐसे बहुत सारे वैरायटीज है जिसको हम लोग इस्तेमाल कर सकते हैं, और उससे भी हमारी उपज बढ़ सकती है, उससे भी हमारी ये जो सहनशीलता है वो बढ़ सकती है। और सरकार का कहना है कि उपज बढ़ाने से लोग धान कम करेंगे और दूसरे दलहन और तिलहन और ये सब चीजें करेंगे। तो क्या हम लोग मतलब धान की उपज के लिए ही लोग धान लगा रहे हैं, या धान की खरीदी होती है, धान को लैंप्स और इस तरह से खरीद लिया जाता है, इसके वजह से वो धान लगाते हैं। तो ये सारे चीजें हम लोग को देखनी पड़ेगी। बहुत सारे हमारे पास इस तरह के वैरायटीज है, जैसे ये देखिए, ये पूर्व भारत के वैरायटीज है धान के। सुंदरबन इलाका के वैरायटीज है, जहाँ हर साल साइक्लोन आता है, समुंदर का पानी अंदर घुस जाता है। वहां पे इस तरह के बहुत सारे किस्में हैं जो नमक को सहनशील, नमक नमकीन पानी में भी होते हैं वो। तो इस तरह के धान हमारे पास है, वहाँ के किसान लगाते हैं। हर स्टेट में जहाँ भी इस तरह की समस्या है, सैलिनिटी का समस्या है, समुंदर का इलाका है, वहाँ पे लोगों ने हजारों हजार साल से बहुत सारे धान उन्होंने निकाले हैं, जो कि हमारा नमक सहनशील है। इस तरह से अलग अलग जो स्ट्रेस टॉलरेंस है, जैसे कहीं एल्कलाइन है, क्षार सहनशीलता भी है, सूखा सहनशीलता भी है, दोनों मतलब सैलाइन अल्कलाइन दोनों कंडीशंस में होते ऐसे धान भी है। फिर सिर्फ पारंपरिक धान की बात नहीं है, मॉडर्न वैरायटीज भी है हमारे पास। सैलाइन रिसर्च के ऊपर, सैलाइन टॉलरेंस के रिसर्च ऊपर इंस्टीट्यूट से करनाल में है, कैनिंग में है, वहां पे उन लोगों ने बहुत सारे वैरायटीज निकाले हैं। तो उनको भी तो इस्तेमाल किया जा सकता है, उनको क्यों नहीं इस्तेमाल किया जा रहा है। फिर बहुत सारे हमारे सूखा सहनशील और अधिक उपज देने वाले पारंपरिक धान है, उनको भी तो लगाया जा सकता है, उनको भी प्रमोट किया जा सकता है। डॉक्टर रिचारिया ने अपने शोध कार्य से बहुत सारे ऐसे धान निकाले थे, और अलग अलग तरीके जैसे श्री विधि है, इससे भी धान की उपज बढ़ सकती है। तो ये सब हमारे पास विकल्प है, इनको नहीं इस्तेमाल करके बस जीन एडिटिंग करना है, जीन को लेके छेड़खानी करना है और कहना है कि सब कुछ सही है, बिल्कुल कोई गलत नहीं हो रहा है। इसकी समझ कुछ माने इतना क्लियरली नहीं आ रहा है, इसमें कई सारे खतरे नजर आ रहे हैं। दूसरी, ये आईपीआर टेक्नोलॉजी है, इसकी जो पेटेंट राइट्स है वो यूनिवर्सिटी ऑफ वियना और ब्रॉड इंस्टीट्यूट, ऐसे सब विदेशी संस्थाओं के पास है। उसके लेके सरकार कह रहे हैं कि हम लोग इसका सुलझाएंगे कैसे, ये पेटेंट को हम इसको कैसे सुलझा सकते, उसके बारे में वो लोग सोच रहे। लेकिन वो भी एक जटिल मामला है कि इसको कैसे वो लोग करेंगे। तो इस तरह के बहुत सारे इसमें समस्याएं हैं, बहुत सारे लेवल पे समस्याएं हैं। इन समस्याओं को नहीं समझ के हम लोग सिर्फ जीन एडिट कर देना है और नए धान निकाल देना है, और ये बहुत बड़ा हमने तकनीकी माने विकास हुआ है हमारा, इसको मानना सही नहीं होगा। और इसके बारे में हम लोग को और माने केयरफुल रहना पड़ेगा, और इसकी जो जानकारियां है इसको पब्लिक में लाना पड़ेगा, तभी कोई कह सकता है कि इसमें वाकई में फायदा है या नहीं। धन्यवाद।
बहुत धन्यवाद शौमिक. I think the most important thing is that gene editing and all applications of gene editing have to be regulated. Government of India should not have deregulated SDN1 and SDN2. When we say to the government that you have deregulated, they come back with an argument that no, there is something called a institutional biosafety committee that is looking at reports. But what is an institutional biosafety committee? It is that committee which is constituted inside a research institute where the researcher is doing this project. Now, why will they, after getting funding for doing this research, will they start saying that something has been seen that is alarming in their reports and so on? This is the job of regulators. This is the job of statutory regulators. India's Environment Protection Act has a definition for genetic engineering. If you read that definition, genome editing clearly constitutes genetic engineering. It should have been regulated by the Genetic Engineering Appraisal Committee, GEAC. But what they have done cleverly is to first deregulate what are called as SDN1 and SDN2 applications, site-directed nuclease 1 and 2 applications, and then announced these two rice varieties as having been readied. We haven't seen the fact that they have tested anything. There's a lot of testing which is not comprehensive enough, not independent enough, not long-term enough in the GM regulatory regime. But even that has not been applied in the case of these genome-edited rices. It is a patented technology, and why should Indian government go and bring such patented seed material and promote it with farmers? And these patents are under exclusive license to big multinationals like Corteva, Monsanto, and others. And Indian government is going to promote farmers of India into growing these rice varieties, claiming this, that, and the other. We do know what kind of rigging of tests that they did to make their claims in the case of GM herbicide-tolerant mustard. The same story could be repeating itself because none of us have seen what are their claims based on. That data, those reports are not in the public domain. Now, after having pushed farmers into these kinds of technologies, IPRs are also, after all, a marketable tool. You can authorize someone, you can even sell patents. And if that is the case, to walk into this thinking that it's a benign thing, when the livelihood security and the food security of this country depends on what kind of seed materials are being used by farmers, why should the Indian government promote patented technologies when the government of India takes a stand in global platforms that India is against patents on seeds and any planting material? It's a contradiction in itself. It's antithetical to the way India's farmers work when it comes to seeds and the way they have used seed as open source material for millennia, for generations and generations. There are questions around seed sovereignty, like Saumik also pointed out. Somebody wrote in the chat box that lots of such products are being used by others safely. No. In the US, despite all the hype about genome editing, only 3 genome-edited crop varieties have been allowed for commercial cultivation, and 2 of those are actually genome editing being applied for Bt and herbicide tolerance traits. Things that can be done through regular rDNA, and things which are problematic with regular rDNA also. What is the new thing that's coming from genome editing? I think everyone in this webinar should understand the complexity and the beauty by which nature has evolved systems by which molecular regulation happens, regulation at the genomic level. And that's the example that Dr. Kritika was trying to give again and again to tell you the complexity. Even though this cell here in my body has the same genome as the cells here, hair is not produced here. Hair is produced only here. Salivary glands and saliva gets produced here and not here, even though the basic genomic similarity or sameness is the same in all cells of my body. The organism is able to make those genes work in particular ways at particular times. It's important to understand that location matters and time matters, and with particular interactions with the external environment as well as the internal molecular and other environments. The complexity of gene regulation is manifesting in what you're seeing in each organism and what the organism is able to do as its functions. Without fully understanding how nature has beautifully evolved this complexity, to think that you have understood such a genome and its regulation, and in ways that are illiterate. The best of geneticists and what they are trying today is actually illiteracy at a higher level. You are doing these changes which are irreversible. With genome editing, the irreversibility is at the molecular level, because whatever you have inserted, whenever it finds that particular CRISPR pattern, it goes on editing it. Nature cannot, natural regulation cannot repair itself, and no copies will be left untouched of this gene, because everything will be altered. Please understand that this is a more irreversible genetic modification than even transgenic genetic modification. And this is genetic modification. There's no point in the government and the biotech lobby packaging it as something else and saying that this is not GM. It is indeed GM. It has a great deal of similarities with transgenic technology. It is in fact dissimilar to genetic changes, mutations that happen with radiation and other kinds of breeding. And it is important to understand, I've been reading a question that was coming up again and again in the chat box, that what are the health implications? What are the health implications? To the gentleman or the lady who's been asking this question, please realize that you will be able to understand if there are health implications only if you do biosafety testing, only if you do comprehensive analysis at the molecular level to understand whether there have been any genome-wide changes, whether there have been any changes other than the ones intended by the geneticists. Today, those tests are not happening. So the very first thing that we should ask, even before we talk about health implications and so on, is to say bring gene editing under regulation, period. Under rigorous, comprehensive, long-term biosafety testing-based regulation. If that is not done, you cannot take this debate to the next level. But even as I say it, I have copied and pasted in the chat box numerous published papers, and these are published in prestigious journals like Nature and Science. Many papers that show that genome editing is unpredictable. There have been examples where, commercial companies have claimed that they've been successful with genome editing in creating something, and then only later, after they get permission and so on, some independent scientists decided from the FDA, decided to relook at what has been presented and found that many changes that have happened have not been reported, have not been tested for. So let's ask for rigorous regulation as the first step. Let's also say, where is the need for such regulation? Where is the need for such technology to be deployed? Are there no alternatives? And thirdly, let's also ask the government, does government, in a democratic setup, do governments have the right, when you and I have a constitutional right, the right to life includes right to health, right to healthy environment and so on, can governments trust untested technologies on all their citizens without any debate, without any questions, by tweaking a statute that already exists, without taking into consideration published scientific literature? We are not saying a thing that is not backed scientifically. You name your question. Somebody was asking again and again, does horizontal gene transfer actually happen, including in human gut? And we will show you studies that are showing yes. In the UK, there's been a study where they have found that the DNA that got inserted in a transgenic GMO that was consumed by the human subjects in that experiment did end up in one of the patients who consented for the experiment. It was found that it appeared as is in the gut. And there is something called horizontal gene transfer with antibiotic resistant marker genes being used, which means that they have the ability to withstand antibiotics. If those genes end up in disease-causing bacteria, your antibiotics, which have already become useless in many cases because of overuse and misuse and abuse, they will no longer be able to control infectious diseases. It's not just the individual human's problem, it is a public health problem. So please understand that there are issues like this, and that is why we are raising this public debate and telling Mr. Shivraj Singh Chauhan that his understanding that GM is hazardous and therefore we have to apply precaution there, but genome editing is precise and safe, he's been misled. They are similar. That's the bottom line here. I have unmuted everyone. If there are any questions, we are willing to take those questions collectively between all the three of us. And there are other participants who are also knowledgeable. Yes, please. Kuldeepak Deshpande ji, can we hear, can we see show of hands from others so that I get an idea of how many are interested in commenting or asking questions, and then how much time do we have? Anyone else who has
Yeah, Kavita ji, I just want to check the possibility that the deregulation of SD1 and SD2, can that be not challenged in a court of law? If the government is trying to bulldoze its way through, um, hoodwinking the public, can we not directly confront them in the court?
Yeah, we can, and we will opt for it after the Janta Ka Court puts pressure on the government. Rushing to courts, for instance, for 20 years, GMO PILs, a whole batch of them were heard by the government, by the Supreme Court for 20 years. There have been some good orders that came out of it, but many benches heard that case. And in the end, it's gone to another bench now when it comes to GM mustard, because there was a split verdict from a two-judge bench. And even the common order is not getting implemented by the government of India, and a contempt petition had to be filed. So yes, we'll go to the courts. But before that, in a democracy where scientific voices are being raised, many of you may not know this, from Nagpur, 20 very senior agriculture scientists, including those who retired as vice chancellors of agriculture universities, have written to the Prime Minister of India saying, why did you deregulate genome editing? So let's have this debate also happening, and we will rush to the court at the right time. Thank you,
And I also want
Kuldeep.
to request, since you are taking so much pain and effort, and both the presenters did an excellent job in trying to explain the nuances of that. But I think there is still opportunity for making it simple as a flyer sort of pitch, which can be understood by moderately educated or even uneducated, or the farming fraternity per se. Because this technical jargon, I mean, it's you have to still apply your mind and get into it in a very micro detail, even for a technical person. But
Yeah.
For laymen, this just goes over the top. And I would
Correct.
really appreciate if we can come out with some kind of flyer where it hits in the Modi language, in a kind of very straightforward rhyming kind of. We need to
Sure.
get poets working on this. We need to get multidisciplinary people working on this.
Sure. Thank you, Kuldeep. For now, if all of you go back from this webinar, understanding that genome editing is not different from GM that many of you have opposed, GM as in transgenic technology, that's good enough for now. But yes, simple flyers, simple graphics, we will attempt that. Suhastai, please go ahead.
Thanks, Kavita. Nice to meet you all after long time.
Yes.
We did successfully stop the GM mustard with big actions, and I think we have to go for something like that. Share your all the concerns you expressed. I was just wondering, because there are so many amino acids deleted, there might be a change of test also and the cooking time needed. I'm just saying this because uh, if those details are known, they could help us to tell other people that see these changes are happening, and maybe even the resistance to pest uh, will also be affected. So for our flyers, like these kind of points may help us um, communicate that there are serious problems even with this technology. Thank you.
Yes, agreed.
Hmm?
I
And
think
wait,
Shaumik
and
did.
would like to contribute to making the flyers if you need.
Lovely. Thank you so much, Suhastai. Shaumik did list down, the environmental impacts, the positive, the possible, impacts on the plant itself, paddy plant itself, from the kind of changes that have happened by the knocking out of two important genes in two genome-edited rice varieties, because they have multiple functions, those particular genes that have been knocked out. Yes, we need a similar list for testing what might be happening to these varieties in terms of human consumption, in terms of human health, and so on. I agree with you. Anupam Da, please go ahead.
Yeah, thank you very much for this beautiful presentation. Actually, I want to add something. What Shomik has told, that I have seen a lot of rice varieties in my life, and still I see a lot of rice varieties in different locations. Those who have developed these varieties, they are not aware of these ecological systems, the rice ecosystem. They have never seen it, I believe. So that's why they are trying to do it in Almira research, or in I think they're in Almira, or in a closed container research. This is not practical research. If I any chance to talk with them, so I can show it to them what rice is. Actually, they are not aware of what rice is, because they have never seen in real situation. So my point is that there are certain salt tolerant varieties available in West Bengal that can give you the same amount of yield, and they are not talking about rice and fish. So when you see rice in waterlogged conditions, you will find rice. So they are not at all questioning, not at all talking about rice fish comparative varieties. In Sundarban areas, I mean, the salt tolerance and the deep water is almost same. So we have certain varieties that can give you 3.5 ton of yield in this locality along with fish. So the total outcome would be more than they think of. So this is the point we should raise. And secondly, that I have asked a lot of my friends in West Bengal, so whether they have seen the trial of these varieties, whether they've seen. Now, unfortunately, I didn't find any except canning. There are several locations that Shomik has told me, that Bankura and Chuchura, the government of West Bengal, the agriculture department, KVK, they said they are not at all aware of these varieties. I don't know when they have conducted this trial. Only Canning Institute, they told they have one variety and they got the trial. So that's why I think that we need, right to information or like that, so that actually exactly where they conducted the trial. We want to know it. This is my point.
Yeah, so the testing or the trials that they're talking about are in fact not biosafety testing. It is, regular agronomic testing in the ICAR's AICRP projects. We can get that information through RTI and so on. But the point remains that we have seen in the GM herbicide tolerant mustard case that such testing is rigged. Data is manipulated, presentation is manipulated, full dossiers are not published in the public domain. So it's a long fight. And we realize that these two rice varieties are basically a Trojan horse to open the floodgates of genome edited animals and plants. And, I just want to repeat what Mr. Jairam Ramesh, when he was the Union Environment Minister, said, which is that this gene technology, I am paraphrasing him, is actually a solution going around looking for a problem. Scientists have a tool. They're now trying to see what is it that they can claim to be solving. I've improved yields. I've done saline resistance with this, that, and the other. So the fact that the problem itself is concocted is an issue. And real solutions lie elsewhere. Is there anyone else who wants to say anything, who is interested in staying in touch? if we can take the liberty of sharing on your email addresses the presentations, we'll certainly do that. Please go ahead.
Does they will written something?
Yeah, we will share the presentations.
Yeah, please.
I see P. Mishra raising.
Hi,
Yes,
am I audible?
yes, you are.
So, my point is, developing countries like India, we have biodiversity, whereas in developed countries, they have technology. They are not aware of kind of biodiversity we have since times immemorial. They are not aware of this many varieties listed, unlisted here in India and in other developing countries. So since they are not aware, they think they are doing something new, very innovative. Such things already exist here in our country. There is no need of modifying the genes. There is no need of modifying or editing um, such genes. I mean, varieties are available here. Indian government should understand this difference, that we are already equipped with such kind of natural, equipped with the. We do have such varieties. We do not need such kind of editing or IP related things. They need money. They just want to, patent all such varieties, and then they will sell their seeds here. This will be draining our economy. We still are agricultural based economy, and we should not allow them to flourish here, to interfere our food things, agriculture things. Apart from health, this is really a big problem for our economy also.
Well said.
Yeah.
Thank you.
And in
So
India,
I request.
yeah.
Yeah.
Yeah, in India, we have patent laws, this 3D section of Indian Patent Act. It doesn't allow such kind of patents, but still they are doing this regulation thing. Just they are trying to find the loopholes,
Yeah.
so that they may take the advantage of this thing. So government should not allow such things. That's it. Thank you.
Agreed. So a request to all the participants here is that, I'm sure you all have connections with some prominent voice or the other. It could be a bureaucrat, it could be a political leader, it could be an eminent individual whose voice is heard. Please think about what is it that you can do. For instance, in the GM-free campaign in terms of transgenic technology, it was state governments which firmly said, as per India's constitution, both agriculture and health are state subjects. You have to consult us and only then move ahead. And the Supreme Court's judgment also, the one that I was just referring to about a batch of PILs, that also, the judgment, the two judges said that. So if you have any space, any ways of engaging with your state governments, please do so. Right now in Tamil Nadu, hundreds and hundreds of people are writing a letter to their chief minister, asking the chief minister not to allow the government of India to bring in genome-edited rice varieties. Please think about things like that to be done in other states also. Mr. Hariraj Singh, please go ahead. And may I request all others to stay unmuted, to stay muted. I'm sorry. Nivedita, please check who's unmute. Okay.
Am I audible?
Yes.
नमस्कार मैम और सभी को मेरा नमस्कार। मैं हरिराज सिंह देहरादून से बोल रहा हूँ। मेरी तो बहुत साधारण सी सोच है जी। हमने जैविक खेती की बात करी, उत्तराखंड में हम जैविक खेती को प्रमोट कर रहे हैं और हम उससे आगे अब केंद्र सरकार प्राकृतिक खेती की बात कर रही है। तो जब आप प्राकृतिक खेती का पूरा फ्रेमवर्क बना के चल रहे हो तो इसमें ये इस तरह का विषय समाहित हो ही नहीं सकता। ये it is actually it is contradicting the basic philosophy of natural farming. एक तो पॉइंट ये है. दूसरा, यदि ये प्लेटफॉर्म और ये ग्रुप इस चीज पे ज्यादा फोकस करे, सोल्यूशन मोड पे, कि भाई ये जो अभी पिछले एक वक्ता ने बोला कि हमारे पास जो सेकंड प्रेजेंटेशन था, इतनी सारी वेराइटीज है जो ड्राउट और सैलिनिटी टॉलरेंट हैं, दे आर प्रूवन वंस. रिछारिया जी ने भी काम किया, तो उनको हम अपने किसी फ्लायर या ब्रोशर में हाईलाइट करें, उसको मास मीडिया में शेयर करें, है ना. तो इस तरह के, फॉर एग्जाम्पल, मैं एक दो पत्रकारों को यहाँ जानता हूँ देहरादून में, तो मैं उनसे इस विषय पे कुछ वार्ता करके वो चीज को पब्लिकली उनके स्पेस में शेयर कर सकता हूँ, शायद वो उसको छाप दें. पर हमें सोल्यूशन मोड देना पड़ेगा. सरकार अगर कहेगी कि ये खाद्य सुरक्षा के लिए ये सब है और बड़े बड़े आंकड़े पेश करेगी, तो उस आंकड़े के सापेक्ष हमें अपने पुराने आंकड़े और पुराने दस्तावेजीकरण को भी प्रस्तुत करना पड़ेगा. तो हम अगर इस तरह का फ्लायर बना दें, सबके लिए कॉमन हो जाए, हम उस पर्टिकुलर कंटेंट को लेके और अपने अपने स्टेट्स में उसकी कैंपेनिंग करें और सब एक साथ मिलके चलें. थैंक यू वेरी मच.
धन्यवाद. Is there anyone else who wants to say anything?
Tirunavakar, sir, Tamil Nadu.
Yes.
Government, under the recommendation of USA, they have introduced a green revolution, but they have not taken the responsibility, and today the agriculture and agriculture are in problem. Now they are going in for second green revolution. There is no guarantee that what it will do for this country. Number 3, America wants to take the Indian agricultural market. Now, if this type of GM and modified products are introduced, the whole products will become contaminated. We should remember that when BT was introduced in Maharashtra, in entire Gujarat, BT spread illegally. Now, if so many products they are developing, it can be a great assault on Indian agriculture. We should stop it anyhow. Thank you.
Thanks. Vijay Kumar ji.
Good evening, madam. Madam, my question is, what is the negative effects of GMO seeds in our body? And my second question is vaccines really changing our DNA? Thank you, madam.
Shaumik, Dr. Kritika. Meanwhile, I'll bring some links for the published literature on GMO effects. Kritika, Shaumik, what are the negative effects?
Yes, so as we have discussed that when we are changing some genes, it may result in changes in the amino acid sequences, it may result in changes in protein, and those genes are having different effects, like they are also contributing to protein and starch accumulation and nitrogen uptake and all that. So it may be affecting the nutrient, it may be releasing some toxins, and it may create allergic reactions in the body. So those are several researches available on these, that how GM crops or even gene edited. Recently there was a gene edited tomato released in Japan, which was having a particular neurotransmitter, and it resulted in increased sleepiness. It was acting like a sedative. So those types of effects do happen, or they are possible, and until we test for them, we don't know.
Kritika, did you want to add anything?
Yeah, like Soumik said, specific examples are also there. And broadly also, I think, like you had mentioned in the chat, Kavita had mentioned in the chat, the possibility of whatever we modify getting into native varieties and completely modifying them, that danger is also there. Through pollen transfer or through horizontal gene transfer, the native varieties should not get affected by the ones that we are attempting by genetic modification.
So, Vijay Kumar ji, there's a lot of scientific evidence. In fact, we published a compilation of such published papers in 2015 last, I think, 2013 or 15. I'll check again whether the link that I shared is the latest edition or there was one more edition. It has different sections, human health impacts, environmental impacts, issues around, molecular level impacts. And please understand that our basis for arguments can be, if we are talking about rigorous science, experimental studies. Because once something is released and you go on asking, did people die in the USA because of GM crops and so on, we'll never know because they've not segregated, they've not labeled. You can't tell whether a person who's died has died because of smoking or environmental pollution or a particular pesticide or GM food and so on. So those variables being many in real life makes scientific evidence being built much more challenging. But in terms of experimental studies, there are a lot of studies which give early warnings, and scientifically as well as legally, including an international agreement that India has signed on for gene technologies called the Cartagena Biosafety Protocol. Precautionary approach is the best way to adopt for governments, which basically, put simply, says better be safe than be sorry. Don't wait for conclusive scientific evidence to prove whether something is safe or unsafe before you take a decision. It is all right to make an error feeling cautious and therefore not adopting a technology because you don't want to be sorry later on. So it's better to be safe than sorry later on when you make a big mistake. In terms of such experimental studies, in the past 10 years, when we did not compile information, a whole lot of new scientific literature got published, which is about both particular genes that are used, how they have toxic effects or allergenic effects and so on, about the process of genetic engineering and thereby consequent health impacts, as well as about chemicals that get used along with GM crops. I'm talking about both insecticides and herbicides. It is actually synergistic effects of all these things that you end up seeing as impacts. In countries like the US, people are pointing out that there is correlation between exponential increase in several illnesses which have become public health problems in the US and the consumption of GM foods. And there have been published papers that compare European public health status, where Europe has rejected GM crops for cultivation, and even when it comes to consumption, if they allow some GM foods to be imported with labeling, consumers don't prefer eating those. And the difference is stark between these two continents, between one large country on one continent as well as Canada and Europe. So there is evidence. If you're talking about genome editing, whether there is evidence, look,
Yes, ma'am.
in the case of genome editing, it is a technology that is about to get introduced into our food chain. It has not yet happened. And therefore, we are saying test first, regulate first. That much has to happen. Why would you hesitate as a aware citizen in demanding that regulation should be there? If you don't want to say no to genome editing, at least ask for regulation. Some of us are very sure that with sound regulation, the inherent lack of safety of some of these technologies is bound to show up in safety testing. Let there at least be testing, let there be regulation. I think we can all agree on at least that thing. Whereas some of us will agree on saying no to genome editing because we have understood what GM itself is. I'll move on to other participants.
Thank you very much, madam, for your kind information.
Thank you. Thanks for joining. Can we move to Jacob? Please wait. Two other people have raised their hands. I don't know how to pronounce GODARR01. Please go ahead.
Yes, I am Mr. Rai Singh Godara from Rajasthan. Okay, and well said by Suhas ji and also by P. Mishra. And I also want to add something, sir, that in India, our farmers are uneducated. Most lot of farmers are, and we need to educate them about the GM crops and their harmful impacts at a broad levels. First thing we want to do, because problem is there, farmers need high yielding varieties, and they don't know are these varieties are hybrids or GM crops are these. They don't aware about all these things. So firstly, we need to educate them, and secondly, our government should not introduce any kind of GM varieties in India. For that, we have you have started campaigns like this, and it should be at a broad levels with a lot of farmers, including with us, and educating them by any type of newspapers or by social medias. There are a lot of alternatives we have to educate the farmers. And one thing is more, if any variety is introduced already, then it is very difficult to again remove from that area.
Correct.
So in starting, I just started two varieties of paddy introducing, as today we discussed here. And at this level, if we are able to stop, then it is much better. And if it introduced, then farmers will adapt it, because they for their economic condition, they also need to be high yielding varieties they need. But which varieties they adapt, is that should be our native Indian varieties or should be
yes,
GM crops. For that, it is needed to aware that. Thank you.
Thank you. Jacob, and then we'll come to you, Jacob.
Hello.
Yes, Jacob.
The issue I think they are trying to do is in intellectual property, just like the BT gene. I've talked to one of the scientists from IRR, and he was saying that they have applied for patent for a particular gene construct. So the whole idea is to make it mainstream, and then technologies, and then make it more lucrative to sell seeds. That's the whole idea, I think, behind this. Essentially, the technology, because it uses
Jacob, we can't hear you. You seem to be in a train. Jacob, we can't hear you. Can you just text? So I am unable to mute him.
Patients and all the essentially.
He seems to be
write in the chat instead.
Yeah, he seems to be talking from a train, but his point was about how we should understand this entire, I mean, conspiracy is one word, but this push towards patented technologies, our farmers and our seed systems towards patented technologies, even public sector scientists applying for patents. So India does not legally under the Patents Act allow patents on seed varieties, but genetic material is getting patented and de facto thereby seed varieties are in a sense patented. This is a story that began with BT cotton in India. So he's raising a red flag on that issue. I saw P. Mishra, you had your hand raised, and I see a Chandrasekhar who's put his video on. Did you want to say anything? We'll close this in the next few minutes. Yes.
Okay, I just wanted to add a small point which already now have been discussed. Since the question was asked, so as a lay person, I would like to add, the person was asking about what are the harmful effects. So as you said, allergic reactions. So we have, bacteria in our gut, so many useful bacteria. And the potential for this antibiotic resistant genes, as you have discussed, and in first presentation also it was discussed, these are used as markers during genetic modification. And there would be horizontal.
Can we not cover things that have already been said? Can we move on to anyone else who has?
Okay, yeah, sure.
Yeah, I mean, we've really, crossed our time limit. We were supposed to have closed this at 6:30, and it's 45 minutes past that time. If there's nothing else, what we will do is stay in touch with you. And in the first mail, we'll ask those of you who don't want to receive mails to say so, and we'll keep you out. We'll share the presentations and the recording link, and let's plan collectively what needs to be done. Is this Zia from HT?
Kavita,
Yes.
it would be good if you could share a draft of writing letters to the CMs. If the content is there, then it gets faster.
So many of you have recently signed on a letter that we'd written to Mr. Shivraj Singh Chauhan, and that letter is present on indiagminfo.org website. This is our official website for the campaign. Please use that as a template, and as soon as we get to creating a template for CMs, those of you who cannot create your own letter, you can use that letter. We will get down to it. The leaflet suggestion, the template letter for writing to chief ministers. we will create those. Okay. Thank you all for joining. You 55, 60 of you stayed so engaged till the very end for nearly 3 hours. Thank you so much. We'll stay in touch. I'd like to thank Shaumik and Dr. Kritika and Harsha for managing the technical parts of this webinar, and Nivedita also. Thank you all. Bye-bye.
Thank you.
Thank you.
Thank you.